So let's recap for a second. Glomerular filtration rate can be regulated, right? So when mean arterial blood pressure fluxes, whether you're exercising or you're sleeping at night, you have three different mechanisms that regulate glomerular filtration rate. You want to keep glomerular filtration rate regular or well constant because you want that urine to flow through the nephron at a steady rate so you can get uh, nutrients reabsorbed and wastes secreted. The first way that we regulate glomerular filtration rate is via myogenic response. This is similar to autoregulation in your arterioles, right? So this means that when your blood pressure goes up, your afferent arteriole automatically constricts. It's a property of smooth muscle. And then when blood pressure drops, your myogenic response of that afferent arteriole is to relax. Okay, that's the weird one. So make a note of this. This is a weird one. It goes against what you think of or what you've learned for your cardiovascular system. The next one is your tubuloglomerular feedback, and we're going to get into this. This involves sodium, and this involves your macula densa cells. It also involves your juxtaglomerular apparatus and the release of renin. And then the last one is hormones and autonomic neurons. And the hormones, renin is considered a hormone, so that kind of overlaps both of them. But then you have norepinephrine and epinephrine as well. Okay. Whoops. Let's see. Let's move this up. Okay, so myogenic autoregulation, the goal is to keep glomerular filtration rate constant. So let me orientate you. Here is your glomerulus, right? The glomerulus is a capillary bed. It's actually a modified capillary bed. Your afferent arteriole is coming into your glomerulus, which is your capillary bed, and then your efferent arteriole is exiting that glomerulus. Notice there's smooth muscle around that afferent arteriole in the picture, right? We have podocytes or foot cells wrapped around this epithelium, well, I guess endothelial capillary bed, this glomerulus, okay? And then what surrounds us? The Bowman's capsule, right? So this is your Bowman's capsule. And then to the right of that is your proximal convoluted tubule. So it's called proximal convoluted tubule because it's proximal to the glomerulus, right? Notice that the Bowman's capsule is simple squamous endothelium, okay? And then this is actually the distal convoluted tubule, and I'll show you a picture of that in a second. But the myogenic autoregulation. So if blood pressure goes up, what's going to happen to glomerular filtration rate? So go to your picture. Let me change the color. I don't know. I screwed up my picture with you guys for my stuff. Well, I guess we'll just, here we go. Let's do green so you can actually see that. Oops. Okay, so that sticks out a little bit better. Okay, so look at the afferent arteriole. If blood pressure goes up, what happens to glomerular filtration rate? That's going to go way faster, right? And so what is this afferent arteriole going to do? It's going to constrict so that glomerular filtration rate slows down or drops, okay? If blood pressure is low, glomerular filtration rate is going to decrease, so your afferent arteriole is going to dilate, and glomerular filtration rate will go up, right? So if blood pressure is low, then glomerular filtration rate is slower, and that afferent arteriole is going to open up or dilate so that glomerular filtration rate goes faster. So that's your myogenic autoregulation. This is intrinsic. This is autoregulation. You don't need to think about it. There's no nervous system input. It's just a property of smooth muscle. Okay. The tubuloglomerular mechanism. You're going to need to spend some time on this. Let me explain this. So your nephron, if you go to the Bowman's capsule, okay, and then the proximal convoluted tubule, and then your loop of Henle goes down, and then you have your distal convoluted tubule. The distal convoluted tubule is smacked right up against the juxtaglomerular apparatus, which is between the afferent and efferent arteriole. Okay, just 1 second, I will be right back. Okay, so the tubuloglomerular mechanism involves the macula densa cells in the distal convoluted tubule. These macula densa cells are sensitive to sodium. So I want you to focus on this green box. Here's your afferent arteriole, right? Glomerulus, efferent arteriole. Smacked in between the afferent and efferent arteriole are the juxtaglomerular cells. If you go through the glomerulus, you have the Bowman's capsule that wraps around it, right? What comes after that? Good, the proximal convoluted tubule. And then you have what? Your loop of Henle, right? Your descending and ascending limb of your loop of Henle. And then you have the distal convoluted tubule. The distal convoluted tubule goes sort of right, it's nestled right in between your afferent arteriole and efferent arteriole. In this region where it is next to the glomerulus, there's these cuboidal cells that are modified. They're called the macula densa. So these cuboidal cells in this region are called the macula densa. They are modified because they're sensitive. Well, they're modified, and the modification is that they are sensitive to sodium levels. So they're sensitive to sodium levels in the urine filtrate. These macula densa cells are smacked up against these juxtaglomerular cells. So these juxtaglomerular cells are in between the afferent and efferent arteriole. Okay. Together, this makes up the juxtaglomerular apparatus. Okay. So let's focus on the macula densa cells first, and then we'll talk about the juxtaglomerular cells and how these guys work together. Okay, so how does the tubuloglomerular mechanism work? If sodium concentration is high in the distal convoluted tubule, that means glomerular filtration rate is going too fast, and that afferent arteriole needs to constrict to slow down glomerular filtration rate. Take a look at the picture. Okay, so orientate yourself. Here's your afferent arteriole, glomerulus, efferent arteriole, right? Afferent arteriole, right? So afferent arteriole is forcing blood through the glomerulus, and then you have urine filtering, or you get a filtrate occurring or filtration occurring. That's number one. And so that filtrate, that urine filtrate or pre-urine, is going to flow through the proximal convoluted tubule, we're at number 2, through the loop of Henle, we're at number 3, and then through the distal convoluted tubule, which is number 4. They're going to go past this number 4, this macula densa monitoring station. Okay, so think about it. If blood pressure is too high, so look at number 1, if blood pressure is too high, what's going to happen to glomerular filtration rate? That's going to go really fast, right? And when that goes really fast, urine flows really fast through the tubes, and urine flows really fast through that macula densa monitoring station. So if there's a lot of sodium flowing past this monitoring station, that means that glomerular filtration rate is going too fast, and we need to slow it down. Okay, so that's going to tell, we're at number five, that afferent arteriole is going to constrict. Okay, pretty cool, huh? All right, let's talk about if sodium level is too low. Okay, so if blood pressure is too low, then flow rate through the glomerulus, or filtration through the glomerulus, is going to be too slow. And then how many sodium are going to go past this little monitoring station? If urine's flowing slowly through the tubes, then not as much sodium is going to go through that monitoring station. And then that macula densa monitoring station is going to say, hey, we need to dilate that afferent arteriole to speed up filtration, okay? To speed up urine flow through the tubes. So that's a pretty complicated mechanism, right? Okay. Let's add to it. So the autonomic nervous system, and then we have hormonal regulation of glomerular filtration rate. So sympathetic nervous system, right? Your neurotransmitter is norepi, your hormone is epinephrine. They are going to bind to, if you look at your afferent arteriole, they're going to bind to adrenergic receptors on that afferent arteriole. And when norepi and epi are released onto that, that's going to vasoconstrict the afferent arteriole. How do you remember that? I think of exercising. So what happens when you exercise? What does blood pressure do? Blood pressure goes up, right? So what would filtration do? Filtration would go up, right? So if blood pressure goes up, filtration is going to go up. And if filtration goes up, you're going to make more or less urine. More urine, good. So when you're exercising really hard, do you pee a lot? Now you might think you pee a lot, but you technically don't. How do you not pee a lot? You're going to vasoconstrict that afferent arteriole so that filtration decreases, right? Remember, when blood pressure goes up, filtration is going to slow down. You want to regulate filtration and keep it constant, right? The hormone angiotensin 2, and that includes the renin, the renin-angiotensin aldosterone pathway, or RAS, right? Angiotensin 2 is going to vasoconstrict that afferent arteriole as well, okay? So those are nervous system, also neurotransmitters and hormones that constrict glomerular filtration rate. Okay, so let's get into what exactly the RAS is. So what does RAS stand for? RAS stands for renin, angiotensin, you can say angiotensin 2, aldosterone system. How does it work? Let's read it and then I'll explain it. So renin is released from the juxtaglomerular apparatus when your afferent arterial map drops. Why? The juxtaglomerular cells don't stretch as much, right? The macula densa cells are tied in. So the macula densa cells sense low plasma osmolarity, or low sodium, or sympathetic nervous system input, okay? So let's take a look. So if you go to the picture, we've already covered the macula densa, the tubuloglomerular feedback, right? So if there's low sodium, then that afferent arteriole is going to dilate so that you get more flow through here, okay? Let's add a layer to this. So if blood pressure is low, these juxtaglomerular cells don't stretch as much. They're actually like baroreceptors. They're baroreceptors, they don't stretch as much, but they're not sending a signal to your brain. They actually don't stretch as much, and when they don't stretch as much, they release renin, okay? So if sodium is low, then the afferent arteriole is going to dilate. If sodium is low, that means blood pressure is low. If these guys don't stretch as much because blood pressure is low, then they're going to release renin. So if sodium's low or if blood pressure is low, either way, you're going to have renin release, okay? So that's why we talk about the juxtaglomerular apparatus, meaning the macula densa cells and the juxtaglomerular cells, because the macula densa cells will trigger the juxtaglomerular cells to release renin. And this is a result of low blood volume, low blood pressure, or low sodium. Okay, so let's take a look at the renin, angiotensin, aldosterone pathway. So blood pressure falls, right? The juxtaglomerular apparatus senses this, or the JG cells sense this. They don't stretch as much, so they release renin. Okay, renin is a hormone that actually acts as a enzyme, as an enzyme. Renin is going to convert angiotensinogen, which is an inactive form of angiotensin, to angiotensin 1. We can say Ang1 for short. Ang1 is going to go to the lungs, and it's going to hit the angiotensin converting enzyme, and that is going to convert Ang1 to Ang2. Okay, angiotensin 2, once that's made, that has multiple effects. It makes you thirsty. So if you're thirsty, you already tried your baroreceptor reflex and it didn't work. So you're moving on to hormonal regulation of blood pressure. So you're already dehydrated. Angiotensin 2 is a vasoconstrictor. So it's a potent vasoconstrictor. So it's going to constrict your blood vessels. And that's going to cause blood pressure to rise. And then angiotensin 2 is going to target your adrenal gland. Do you remember what zone of the adrenal gland that releases aldosterone? It's going to be the outermost layer, the more superficial, the zona glomerulosa. So the zona glomerulosa is going to release angiotensin, the zona glomerulosa is going to respond to the angiotensin 2 by releasing aldosterone. Aldosterone is going to go to your distal convoluted tubule. It's not enough to say kidney or nephron. It's your distal convoluted tubule, and that is going to cause you to reabsorb sodium, and water is going to follow it. So your blood pressure is going to rise. Okay, so here is a little animation video about renin. So I clicked on the link from the PowerPoint slide.
It is important for the body to keep sodium and water balances in check, and also to maintain healthy blood volume and blood pressure. The renin-angiotensin-aldosterone system assists with this. When the glomerulus, a bundle of capillary blood vessels found in the kidney, senses a drop in blood flow or sodium, it secretes an enzyme called renin into the bloodstream. The renin moves to the liver, where it converts the inactive peptide angiotensinogen to angiotensin 1. Angiotensin 1 travels to the lungs, where another enzyme converts it to angiotensin 2. From there, angiotensin 2 makes its way to the adrenal glands at the top of the kidneys, where it stimulates the production of aldosterone. Aldosterone helps the kidneys conserve sodium and water, leading to increased fluid volume and sodium levels. If blood flow to the kidneys or the amount of sodium increases, less renin is produced in an attempt to normalize blood pressure.
Okay, so we're at the renin-angiotensin-aldosterone system pathway, or RAS for short. I wanted to take a second to talk about the coronavirus and how there's some research out of Italy. It hasn't been peer reviewed yet, but there's, everybody's rushing to get out research so that clinicians can help, treat patients. So let's look at the angiotensinogen. So we'll start at angiotensinogen, right? That's at the liver. Renin converts that to angiotensin 1, and then angiotensin 1 gets converted to angiotensin 2 by way of the lungs via angiotensin converting enzyme, right? Or ACE for short, because it stands for, the abbreviation is ACE, right? Angiotensin causes vasoconstriction. Angiotensin 2 causes vasoconstriction. There's also an angiotensin, and I'm going to write on this. Let me see if I can get this to work. Okay. There's also pen. Once angiotensin 2 vasoconstricts everything, there's going to be, and this occurs in the lungs, right? This enzyme is attached to epithelial cells in the lungs, epithelial type 2 cells. Um, Angiotensin is going to cause vasoconstriction, but then actually, maybe we'll do it over here. It's going to get converted to angiotensin 1,7. And I'm not obviously going to ask this on an exam, but just so you have an idea of how this works. And this happens by ACE2. Now, Ang 1,7 is a vasodilator. Okay, so it shuts down the vasoconstriction pathway. If we're regulating blood pressure, we can hit the angiotensin converting enzyme or inhibit the angiotensin converting enzyme from making angiotensin 2, or we can have something that binds to ACE2 or angiotensin converting enzyme 2 that's going to make more Ang 1,7 and vasodilate blood vessels, etc., and drop blood pressure. Okay. Now, ACE2 is on the surface of, again, those epithelial cells in the lung lining, and this is the target of the coronavirus. So the coronavirus, the spikes of the coronavirus, actually bind to this ACE2 enzyme, and because this enzyme is on the surface of those endothelial cells, and they actually form vesicles. So the virus, right, the coronavirus plus the ACE2 get pulled into the lung lining, the epithelial lung lining. And that's how the COVID actually enters your lung lining. And that's why the COVID causes so much destruction to that lung lining. It's also why people who regulate blood pressure through this pathway are more susceptible to the COVID virus, because when you take blood pressure meds upregulate this pathway. So you have more enzymes at that lung lining means more chance for COVID to jump in and attack. And so it's really dangerous. So I just wanted to give a shout out to Professor Tatiana Vasquez, for sending that to all of us. There's so much research, and none of it's peer reviewed yet because nobody has time. They're just trying to get this out to the masses. But it's coming out there, and it'll be interesting in the next year or two how much we learn about this novel virus. It's really interesting from a scientific standpoint. Okay, let's see, moving on. So glomerular filtration rate can be estimated. Let's say I have a person, or I think that my glomerulus has been damaged, and I want to see that what my glomerular filtration rate is. We can look at creatinine clearance rate. Okay, so we can look at volume of blood that's cleared of creatinine, which is a waste product by the muscle, or from the muscle, over time, right? So creatine is secreted as well, but we can block that by cimetidine, right? What are we interested in? So let me back up. So when you go through metabolism, you are making urea and creatinine. Those are your waste products. Those get kicked out by the kidney, it gets filtered out through the glomerulus and kicked out, secreted from the proximal convoluted tubule. When kidney disease is suspected, they're going to look at creatinine, should say creatinine, and blood urea nitrogen in the bloodstream. And so if I have a patient who has kidney damage, or I suspect they have kidney damage, I'm going to take a blood sample and see how much creatinine and blood urea nitrogen are in their bloodstream. The idea is the more that the glomerulus is damaged, the less of these are filtered out. And so if they're not filtered out, they're going to stay in the blood. So the more damaged the kidneys are, that results in more creatinine, right? And more blood urea nitrogen, or BUN, in the bloodstream. Why? Normally, when there's no damage, right? So when there's no damage, these guys get filtered out and they're in the urine. So you can measure the urea and the creatinine in the urine. When the glomerulus is damaged, when that endothelial lining is damaged, the more damaged it is, the less waste products get filtered out, and the more of those waste products stay in the bloodstream. And so clinicians can tell how much damage there is of the glomerulus based on the levels of creatinine and blood urea nitrogen in the bloodstream. When do we see damage of the kidney? I would think of diabetes mellitus. So when somebody has diabetes mellitus, the sugar acts like sand to capillaries and starts ripping apart the capillaries and causes scar tissue to capillaries, among other things. And if there is scar tissue, then the glomerulus can't filter the blood properly. Other things with diabetes mellitus would be neuropathy, right? So the sugar is taken into the nerve cells, and that prevents the nerve cells from submitting signals. Likewise, because you have high blood sugar, the glucose is going to go into the red blood cells and get glycosylated, and so you're going to have a higher amount of glucose in those red blood cells, and we measure that, well, glycosylated glucose, I should say. We measure that using A1C tests, or we call it the A1C test. And we'll talk more about diabetes mellitus when we get to sugar handling, okay? All right, so reabsorption, secretion, and excretion. Let's talk about the different ions, right? So sodium and water, what do we use sodium and water for? Extracellular volume and osmolarity, right? Particle concentration. Potassium is important with cardiac and muscle function. Calcium is important with muscle contractions as well, exocytosis and other functions. Protons and bicarbonate regulate your pH. Your body must maintain mass balance. What the heck does that mean? Whatever you're taking in, you need to kick out. So if you're taking in excess protein, you need to get rid of the nitrogen source, and so you're going to kick it out at the kidney. If you're making more CO2, you need to kick that out of the lungs. You need to regulate pH at the lungs and kidney. Okay, so this is a busy slide. Let me go over it with you, and then I'm going to actually draw my own for you so that you let's see what color, I guess we'll do blue, so that this makes a little bit more sense. Okay, so let's take a look. So this is your nephron, right? Here's your proximal convoluted tubule, your loop of Henle, distal convoluted tubule, and collecting duct.